My name is Nick Keeper. I'm the Executive Dean of the School of Science, and welcome to the latest of our Science Week presentations. The presentation this afternoon is by Professor Bonnie Wallace on sodium channel structures and cardiovascular and neurological diseases. I first met Bonnie, I think, at a British Crystallographic Association summer school in the early 1990s, if not a little before then. So I've met Bonnie for quite a long time. We also were both alumni of the MRC Laboratory of Molecular Biology in Cambridge, but Bonnie's been at Birkbeck for around about 20 years and is currently leading our membrane protein work. So she's going to tell us about sodium channel structures.
Can you all hear me without the microphone? Good, because that means I can walk around, and I like to do that. I'm here today to give you a bit of an overview of what structural biology can do for you, or hopefully a collective us as a population. Structural biology is a discipline within the biological sciences that lets us actually visibly look at molecules, and by looking at them, hope to understand how they work and how they function, and ultimately helps us to understand what goes wrong in diseases and how we might make new drugs that will help solve those diseases for people who have them. So today, I'm going to focus on a protein structure that we've worked on for many years. And for many years, I mean more than 10 years, this has been the focus of my lab. And you will see when I go through a timeline that this is not a short and easy thing to do. It's a long-term commitment, but hopefully the rewards turn out to be very useful, both in academia and ultimately for the pharmaceutical industry for developing new drugs and for the populace who are going to have those drugs to treat various diseases. And in this case, sodium channels are very important because they cause a number of different diseases, including cardiovascular and neurological diseases. So ventricular fibrillation associated with heart attacks, neurological diseases such as epilepsy, and one that many people have, chronic pain. And so these are relevant to us in our lives. So what is a voltage-gated sodium channel? Well, it's a membrane protein, and membrane proteins are found in the outside of the cell. So this is a really simplified version of a cell in which the cell has all of its important contents, its nucleus, ribosomes, and all of the functioning organelles inside. But it's surrounded by a membrane. This is a semi-permeable type of material that lets important things into the cell but keeps things out of the cell also. And maintaining the balance across the cell membrane is very important. Some things don't naturally go through the cell membrane, and those include ions. But ions, such as sodium, so sodium is all around us, the seas have a high concentration of sodium, and maintaining the balance of sodium inside and out of the cell is absolutely critical for the cell to live. And the kinds of molecules that transect that membrane, so here's a piece of that membrane taken out, and look at it sidewards, are things that are called ion channels. And those ion channels specifically let certain ions in and not other ions in, and prevent ions from going out. So the one we're focusing on is the sodium channel. So the sodium channel enables the movement of sodium ions from the outside of the cell through this cell membrane to the inside of the cell, where it's absolutely important for functioning. The opening and closing of sodium channels, so these undergo, as we'll see in a minute, an opening and closing function, is absolutely and carefully regulated, so there isn't any leak of sodium. So leaking of sodium either in or out of the cell in a way that the balance is not maintained would kill the cell. So we have to keep the right amount of sodium inside and outside. The way that it normally operates is that based on the voltage across the cell, it opens and closes appropriately. But sometimes the opening and closing can be stopped by specific toxin or drugs. Drugs which can help regulate it if you're not opening and closing it at a correctly timing, or toxins such as we're going to see in a minute from a variety of organisms that from their name are toxic to cells, and they act by killing the cell by either keeping the sodium channels open or closed. So here's a diagram of what the sodium channel was thought to be looking like for many years, up until about a year or so ago. And is embedded in this phospholipid membrane that surrounds a cell. So here's the cell around here. And this continuous membrane keeps sodium ions out normally because it's very hydrophobic and sodium ions are hydrophilic. The ions go through the cell in this regulated manner. There are different versions of sodium channels found in all animals and in some bacteria. They're all very similar, and that turns out to be both interesting and a problem. They enable the transmission of electrical signals along nerve fibers. So when you sense something and you send an impulse either to or from your brain, that is the result of electrical signals along the nerve fibers. And the first step in that signaling process is called an action potential, and that's when these particular channels open. They're also found in bacteria, and they enable bacteria to swim around and live in extreme environments such as thermal springs. Normally, a cell doesn't want to have very much sodium in it, and if you're living in a thermal spring which has very, very high salt concentrations, if this opened in the same way as it does in human cells, what you do is allow lots of sodium in and effectively kill the cell. So there has to be a careful mechanism in thermal springs and other extreme environments so that these don't allow the bacteria to take in sodium ions. But in humans, sodium channels are responsible for neurological and cardiovascular diseases and the targets now for a number of drugs that are currently on the market and are being developed more explicitly for the future, such as anti-epileptic drugs in the brain, analgesics, such as local anesthetics used in, for instance, surgery as an anesthetic, or antiarrhythmics, so people who have arrhythmias in their heart or irregularities in their heart rhythm may be taking drugs that affect sodium channels. So electrical signals that are transmitted along nerves are due to the opening and closing of these channels. And at different voltages, you either keep the channels all closed, or sometimes they open and then they close and then they open and they close. And these are just simply electrical recordings of showing what's happening when these cells open and close. And we're very interested in understanding how they open and close. So there have been models for how this opening and closing action occurs for many years. And here's one of the most common ones that was produced several decades ago now, in which the sodium channels were believed to have three different states. The resting state, which is the closed state, in which if this is the sodium channel, there's a paddle here that opens up and a paddle at the top, and these two flaps prevent sodium ions either going into the cavity and then going across, or going into the cavity and getting stuck and not going out. So the closed state has a paddle across the top, and the open state has both paddles open, and the inactivated state has it closed but with the paddle at the top open and the paddle at the bottom closed. As we'll see, hopefully at the end of this talk, this was a really nice model for how things work, but turns out not to be how they actually work now that we know what the molecules look like. But it's a really good model as a context for understanding what we wanted to look at. So we can artificially open and close the cells in ways that nature does this too, because there are a whole bunch of naturally occurring toxins that bind to sodium channels and either stop them opening or closing them, or can keep them in permanently open states. And so there are a number of them in organisms that may actually look fairly familiar to you, but which you shouldn't be touching, for instance, because if you open your sodium channels or close them inappropriately, what it can do is stop your heartbeat or your sensations of electrical signals. So one of the most common is the fugu fish, also known as the blowfish. For those of you who eat sushi and eat it in upper-end restaurants that serve this is a particular delicacy, you hope that the chef is very good, because he dissects out the small amount of the material that has the toxin in it. And if he gets it wrong and you get the toxin, there are many documented cases of people dying from eating fugu fish. Poison dart frogs, don't come into them as commonly as you might. These are often in the Amazon regions. Scorpions, you really hope you don't encounter, or cone shells, which are mostly found in the Australia-Asian seas, all of which produce different kinds of toxins that have different effects. Some of them now are turning out to be quite useful for making drugs, and in fact, cone shells produce a kind of toxin that is now being used as a mimic to treat severe and chronic pain. So they aren't all bad, but they all affect sodium channels. How we know they cause these different conformations that I said earlier, the resting, open, and inactivated states, are by looking at different methods that can tell us about the structure of the protein, very indirectly. And this is a method we use in our lab. It was developed a number of years ago called circular dichroism spectroscopy. It doesn't really matter what the method is. What you can see is that there are different traces, the red and the black, and they correspond to two different states, the open state and the inactivated state. And by looking at the spectrum of these patterns, we can tell whether or not the channel is in its open or closed state. We can also make it go to one of the states, so we can take a batrachotoxin that is one of these naturally occurring toxins and make the channels always closed. So you never let sodiums in, which is not a really good thing because you never transmit them. Or you can take something like lamotrigine, which is an anti-epileptic drug. And what its role is to keep the protein always in its inactivated state. And we can see that the different states can be differently depicted by this method. But that isn't terribly satisfying, just to say we can identify them. We'd really like to know what they look like. So our goal has been to look at the molecule in atomic level detail so we can see exactly what it looks like. So first of all, we need to know kind of what the overall structure of the molecule is. And this is a diagram of what a eukaryotic or a human voltage-gated sodium channel looks like. It consists of 4 different domains, all of which are very, very similar. They each have, within the membrane segments, 6 transmembrane segments. S1 through S4, which form a voltage sensor, and S5 and S6, which form the pore. These fold up in a way that has always been depicted as looking at bundles. So this is a bundle, and this is a bundle, and they all form together to form 4 bundles that have a hole down the middle that allows sodium to go through. And this is based on the sequences of the molecules, and has been our closest view of what the structures must look like for a number of years. In fact, we can divide them up because each of the domains is made out of these 2 subdomains, this pore region, which forms the place where the sodium ions go through, and the voltage sensors on the outside, which actually regulate the opening and closing of them. So people, for more than 20 years, have been actively trying to crystallize and solve and determine the structure of human or eukaryotic sodium channels, but they're just too hard to do. And we and probably 100 labs around the world have been trying to do that. But fortunately, fairly recently, it's been found that there are prokaryotic or bacterial sodium channels that are almost identical to human sodium channels. And they turn out to be easier to handle, or easier on a large scale of hard. The people in my lab that have spent the last 10 or 12 years doing this think that they're not particularly easy, but relative to the large bacterial channels, they are. And there are more than 30 of them from different kinds of bacteria, mostly found from thermal springs and other extreme environments. And they're highly similar to the eukaryotic or human channels, with more than 30% of them being identical to human channels and having very similar structures in other ways that we can look at them. Furthermore, their functions are very similar to human channels, which is good because if we're going to look at structure and function, that's important. And they bind eukaryotic channel blockers, so they're blocked in the same way, so these toxins can also bind to them. And we now have the ability to produce and purify them in large quantities so that we can do structural studies. That's one of the problems of looking at in minute detail at the structure of molecules, is that in general you need quite a large amount of them to be able to make quite small crystals. So prokaryotic sodium channels, why they're easier, is instead of 4 of these domains, they consist of only a single domain, but nature makes them 4 of them join up to make a tetramer that looks essentially identical to the tetramer that was one molecule in a human sodium channel. And in fact, the channel in the middle of them, which is what we're most interested in, this pore, looks very, very similar. But it's now from 4 different polypeptide chains. And they also have a voltage sensor. So I thought I'd just give you a flavor of the process of determining of the molecular structure of this sodium channel and the kind of time scale that it takes to do this. And the hard work that many students and postdocs have done to achieve this. So first of all, the first process that you have to do is you have to isolate the gene. Often you can do that from looking at the gene sequences. So in the Human Genome Project, we have the sequences of all the genes in the human body. We have that for many other organisms, and we can find that gene and isolate it, and then use it to express and purify the channel. So we can purify this channel. An indication that we've purified the channel is that if we run something called an SDS page gel, which is simply a way of purifying one protein from any others. The reason that we know this is pure is that there's one band right there, and it's big, and that makes us very happy. This slide is the combined work of 3 PhD students and 1 postdoc over 6 years. So I can say it's now easy to purify these things, but then it wasn't me that actually did them. And over the process of doing this study, we started back in about 2000, and we got the gene for the channel by 2003, and we expressed and purified the channel by about 2005. Then the next step is to grow crystals. Crystals are the way that we can use to look at the proteins by using X-ray diffraction. Now, these are the first crystals that we made of this sodium channel, and you can see by the scale here that the size of these is really very small. They're 300 microns or less. So a micron is a millionth of a meter. So you can see they're really, really, really tiny things. And we're going to shine X-rays at them. And the X-rays are going to let us look at the internal structure of what the molecules look like. And you can think of it like shining an X-ray when you go to the doctor. So you get a chest X-ray, and you stand there in front of the X-ray machine, and they take a picture, and behind you is, you see effectively an image of your bones, or something that has a high density in them. What we want to do here is effectively do the same thing, except that because these are really, really small, if you put the same amount of X-rays that you put through a person, you wouldn't see anything, because a person has a lot of material there to stop and interact with the X-rays. So if we think of these as being only 300 microns, we then need a really, really big X-ray generator. Not like the kind you can see over at UCLH when you're going to get your chest X-ray, but effectively something called a diamond synchrotron. This is in Oxfordshire, and the UK has one of these, and several European, American, and Asian countries also have synchrotrons. And they are an enormous source of X-rays. And so they'll put in an awful lot of X-rays into the sample. Orders of magnitude more X-rays than your chest X-ray. But that is good because the crystals are orders of magnitude smaller than you are. Just to give you an idea of the size of things, that's a big van down there. So this is an enormous facility that generates X-rays that go around the ring, and you actually put your tiny, tiny crystal. So this is this really small thing. You put it in this big instrument, and out come X-rays. And if you're lucky, you get spots. Well, this was our first spot after 2 or 3 years of trying to look at these crystals, and it was really a major disappointment. The more spots you see, the better off the structure is going to be, and the better the crystals are. And if you look really hard, and we as biophysicists look really hard because we want to see them, you'll see a small ring of spots there and a couple around here. This is not good. This is a very bad crystal. And so, over the course of the next 3 years, we worked to get this. It's a little dark, but what you can see is there's rows and rows of spots all over the place. And these mean that these crystals diffract really well. So, after crystallizing the channel, in 2007, we got our first X-ray diffraction pattern, that one with a few little spots and the eye of faith that told us there were spots there in 2009, and the first good diffraction pattern in 2011. And that's not the end of the story. The story then continues with the process of solve the structure, which involves a lot of physics and mathematics to calculate how that pattern of spots relates to where molecules or atoms are in a molecule. And then finally last year, we actually succeeded in solving the structure. So as I was going to show you, I was just going to wave my hand and say, and we solved the structure, but I wanted you to get a feel for what people went through in the lab to get this far. So we're talking about a decade or more of work to produce a structure. And this is what the structure looks like after all of that time. So here's the membrane, and here's the molecules. Now I said there were 4 molecules that were identical in the structure. So I've made 3 of them gray because we're only going to look at one of them. But what they consist of is, here's a helix that goes through the membrane. Then there's another helix, this yellow helix, that comes down here. And then there's something I've labeled SF, which is the selectivity filter, which we'll see in a minute is the all-important thing that chooses between sodium and potassium and calcium ions for this. And then it follows on through to the other end of the membrane. And there's this kind of cavity up here where sodium ions come through, bind in the selectivity filter, find another cavity around here where they slosh around in some water, and then pop out the bottom. So that's what the overall structure looks like. The external surface, so this is what's called a ribbon diagram, and so we're only looking at the overall backbone fold of the molecule. This is another way of depicting it. This is what the surface of it looks like, and we can see here it's all quite white. White means that it's a neutral charge. It has no positive or negative charges except for a few red and blue spots for the positive and negative charges, except for at the ends. You can't see this end very well, but you will in another picture, in which we have, because they're going to let sodium ions through, they tend to be negatively charged so that they attract the sodium ions. So our interest was the differences between the open and closed states, because as I said, the first step in the action potential in nerve conduction is this opening process. How much does the molecule have to do to open and close? Well, we can compare the difference between open and closed structures by the red and blue diagram, ribbons. And what you can first of all see is that they look pretty similar because most of the places they're overlaid. And that's the important thing, is that this molecule doesn't change very much to open and close. How does it do it? Well, here if we're looking at the external surface, this is the top of the pore, and there's the hole down the middle, and you can't really see much difference at the top. If we look at the bottom of it, this is the inside of the cell, there's a small motion corresponding to this yellow arrow for each of the 4 monomers in the tetramer. And what that does is it opens out the hole in the middle to make room for sodium ions. How it does it is that one residue in the middle of one of these helices moves. So it goes from the red position to the blue position. But the blue position is the closed position. And what the closed position does is it, just as the sodium ion is going to try and go through there, it hits this blue position and it can't go through. It can only go through if it's in the red position. So that's not really very easy to look at. So I've made a little bit of a video showing the morphing between the open and closed states. And what you can see, what I thought you could see, is, I didn't test this one. I tested all the other videos, but what you could see if this opened and closed is that these actually open and close like this to form sort of a clasping motion, and that's the bottom of the molecule. This is kind of how you can represent it simply. It's like the opening and closing of a lens. So the hole is big when it's open, and then there's a twisting motion, and it closes up like the end of a lens. And this is too small a hole for the sodiums to get out, and this is big enough for them to get out. You can think of it looking at the end of it. I told you that it was negatively charged and very red at the top. Here it is. And there's a hole down the middle in the open form where the sodium ions can go through. And there's a plug on the same place in the closed form where the sodium ions can't go through. If we cut a slice through the middle of it, you can see there's a hole that goes all the way through there, but it hits a plug when it gets to this one. And so this is the closed state and that's the open state. And here's the continuous hole. This is a surface showing where the ions can live in the channel. There's lots of room up here, quite narrow in the middle, and then it gets wider and wider till it goes out the bottom. Now, here's the movie I think is going to work, of showing you the dynamics of this molecule. Hopefully. Yes. And this is what shows you, this is a sodium ion, and this is what we're going to watch it go all the way from this surface to this surface. Now much of the time it sits around and just juggles around and doesn't do very much. This is what's in the selectivity filter that chooses between the sodium and any bigger ions. You can see it's going to almost make it now, and there it goes out the bottom. And if we wait long enough, we'll see another one come in and go out the bottom too. So it's not, you might say it doesn't look like a very fast process, but these are on the order of picoseconds. And we're looking at the total of 1 nanosecond of time that's going by in order to watch this occur. These little blue things are water molecules that can go back and forth as they want in the channel. Sometimes there's a counter ion that goes through, that's the little green balls, but eventually there's a net motion of all the sodium ions that go through. So if we had looked at the closed form of the molecule, the plug at the bottom would have prevented any of them to go through at all. Hopefully, go back here. So that was the skill shots in case the movie didn't work, but it did. So you've seen what it actually happens. And we can predict and measure the conductance parameters. So we know that it's now selective for sodium over potassium by a ratio of about 20:1, which is good because we don't want it to let potassiums go through, because the potassium enters the cell later in the action potential. And that means that if you had it enter then, it would mess up the nerve conductions. And the conductance is just about what we predict. So the changes in the structure that occur between open and closed are all at the bottom end. So this is what's known as the selectivity filter. That's what chooses between sodium, potassium, calcium, and magnesium. And they're all the same in the two states, the open and closed forms. It's just down at the bottom that there's an opening and closing. So that sodium ions can happily enter here, or they could happily enter here, but they can't get out the bottom. But if we look at the closed state, this is an end-on view of it, and this is the hole that they're going to go through. You can see the hole is empty. But when we look at the closed state, here's the same molecule and the same part that it's going to go through, but now there's this big blob of what's green here, and that green turns out to be a sodium ion. And so we can actually very clearly say where the sodium ions fit in the middle of this selectivity filter and where all the waters reside during the process of going across the membrane. And we can find where the ions are in the pore, either looking along the membrane surface or down into the membrane. But the selectivity filter is specific for sodium and not potassium, because we know the structure of potassium channels, and they're quite different. So the sodium channels are the red and blue here, and the potassium channels are the yellow. And what you can see is that the hole in the middle here, where the ions bind, is really, really narrow for the potassium ions, because they go in without water bound around them and are smaller. And so because of that, sodium ions are not allowed to go through these channels. And that's very important, because the cells all have potassium channels as well as sodium channels, but you don't want sodium to go through potassium channels and vice versa. So then the last question is the last state, the inactivated state. Well, in this case, there are many differences in the selectivity region, and the selectivity region basically collapses and forms a blockade for ions going through there, but not down at the bottom of the molecule. The last part of the molecule we haven't looked at here is what we call the C-terminal domain. This is the end of the molecule that's beyond the crystal structure. So here's the crystal structures we know about, but somehow this part of the molecule isn't seen. And the question is, why don't we see it, and do we care if we don't see it? Well, it turns out we do. because this part of the molecule is essential for fully functioning molecules. So full-length molecules conduct, this is the conductance of sodium ions, and ones that are missing the C terminus are flat and they don't conduct sodium ions. So we wanted to use a method to look at what the end of the molecule was, because we couldn't see it by crystallography. So the first method we use is a method we developed called Synchrotron radiation circular dichroism. Uses that same kind of big machine that looked like a football field, but uses a different wavelength to do measurements. And we can measure differences between these molecules, and what we can do is define the structure of that part of the molecule as having disordered and helical parts of the molecule. And then we can use, and this was a collaboration with UCL, we used a method called electron spin resonance to define the nature of this part of the molecule. And the nice thing about it is that, if I can get this movie to work, I'll show you that part of the molecule is very flexible. And this is what the molecule looks like. So here's the part where the sodium ions go through, but there's this bit at the end that's going to bobbing around and weaving around. And that motion turns out to be important for how the molecules actually function. So you can see that it never stays in one place, and that's really important for how they work. Back to this. That's the static picture of what I've just shown you in the movie. And in fact, this is our crystal structure in which the moving parts are placed in the middle of it, and we can see that they all line up very nicely. And now that gives us a new and the first hypothesis for the opening and closing action of these channels. So there's a closed pore, and here is that C-terminus part, and it's got a little weight at the bottom of it. And when the channel opens up and these parts of the molecule move out, what they do is they pull this part of the molecule apart, and without changing the little weight on the end, we've got kind of an oscillator motion now that goes up and down and opens and closes without unfolding the molecule. If this unfolded, it would be very costly because the whole thing would fall apart. So we want to hold it together, but we want to allow it to flex. And that's what the new hypothesis is for its activation. And now we have a complete cycle: the closed state, the open state, and the inactivated state. So we can look back at the original model and see how good it was. Well, actually not very good at all. All three of the states turn out to be wrong. But we can now start to say what is correct. So we have a closed state now here. But in this state, we've got the selectivity filter, so the ions can go through, but this part of the molecule is closed by this little narrow closing mechanism. In the open state, there's a slight flap that opens here, and that allows the sodium ions that have already entered in the resting or closed state to go all the way through. And then during the course of inactivation, these flaps close up here, but also the top part where the selectivity filter comes together. So now we have the top part closed and the bottom part closed, and then in the process called recovery, which is part of the nerve conduction process, these come apart, but these stay together. So we have bottom opening, bottom closing, top closing, and then bottom closing and top opening. So we now know what all the states are, simply based on the structure we've been able to see. And for us, the exciting part was to see the structure, to actually not hypothesize with a lot of little moving pictures like this, but to actually see a picture of what the molecules look like as they change. I talked about this to a group of artists a couple months ago, and I explained the process of scientific discovery as the thrill of looking at something and knowing you were the first person ever to see what that structure looks like and how it might work. And I just get a real thrill. And the artists told me that they get the same kind of thrill when they're doing and making new pieces of art. And they actually we're quite interested to see that science and art aren't all that far apart, because it's all in the inspiration and thrill of what you're doing. And if you're not interested and thrilled by the science, then probably science isn't for you. So, having done all this, what does it tell us about human disease? Because really that's what we're really interested in. You know, it's very nice for these bacteria to swim around in thermal springs and stuff, but, you know, who cares? But human disease we do actually care about. So here's an example of what we can tell now based about human disease and some diseases. So the sodium channel, called NAV1.7, it's a very creative name. They're all called, there are 9 of them in humans, and they're called 1.1, 1.2, 1.3, and you get the pattern. But 1.7 is the one that causes pain. And it causes pain, and we know it because you can see at least 2 kinds of different diseases that involve it. The first one I'm going to talk about in a minute is called erythromalgia, which is also known as burning foot syndrome. And these people have sodium channels that are effectively on all the time. They never properly close. So their neurons are firing and they're getting nerve impulses all the time. As a result, their whole body hurts because they've got pain all the time. It's most predominant in their feet, hence the burning foot syndrome, but it's all over. And it's a really very painful disease to have. On the other hand, there's another kind of disease called the loss of function disease, in which you make no NAV1.7 channels, so you don't have any stimulation at all associated with the nerves. And these people feel no pain. This isn't a trick picture, you know, like you sometimes see in the circus or whatever. This man actually could, if he was careful and not hit any of his internal organs, place swords or anything into himself without actually feeling anything. People have been known, it's most manifest in families of people that go to A&E, and they go because they've got very bad burns on their hand, because they put their hand on the hob and they haven't realized it's hot, because they don't feel any sensation there, and they get horrible burns. So you might think, particularly if you've got chronic pain, it'd be really good not to feel any pain, but actually not feeling any pain can be very dangerous too. So both of these are important diseases. And just to tell you a little bit now, we know the molecular nature of this disease because of knowing the structure. So that's caused by a single mutation in one of the domains of the human channel, and it's a very tiny change from a slightly bigger residue to a slightly smaller residue. And all of the sodium channels that have been known in humans all have the F, which is the bigger residue, but except for this mutant, which is the smaller one. And what does that do? Well, here's the wild type, or the normal gene. And what normally people have are these, this is the very bottom of that sort of funnel-shaped thing I showed you. And there are these what are called aromatic amino acids that are very hydrophobic and form a plug at the bottom. And you can see them here. There's 4 of them, and they form effectively a plug, and this hole in the bottom isn't big enough for sodiums to come through. In fact, they can only come through when it does this motion of opening and closing. But in this disease, what happens is people have 3 of these part of the 4 plugs, but not the 4th. What you can see here is there are the 3, and this one's missing. And what it means is there's a big hole down the middle. And that big hole down the middle means that sodium ions can go through even when the channel doesn't open. In its closed state, there's a hole big enough there for them just to run through. So those people have sodium channels that fire all the time. So here we have a basis now for understanding a human disease. It means now that we can start to do rational drug design of pharmaceuticals to target sodium channels, to open them or to close them, depending on the disease. So whether you have a disease in which they're never open or they're always open. Drugs must bind tightly to be effective, and they must be specific for the target tissue to eliminate side effects. As I said earlier, it's very important to know that there are a number of sodium channels in the body that are found in different tissues. And what you don't want to do is treat somebody for epilepsy and then give them a heart attack. That actually isn't a very effective drug. So you need to be able to make a drug that very specifically hits one of the sodium channels. So noting the structure of the sodium channel will help us to design such drugs. And here's an example. This is lamotrigine. It's an anti-epileptic drug. It's the most commonly used one now on the market. It's very effective. It binds to sodium channels. We wanted to understand how it would bind to sodium channels. This is a model for the sodium channel, and we're going to see how it will fit in there. Eventually, we will actually solve the structure of it with this bound to the sodium channel, but for now, we're going to try and see if we can understand how it might bind. Now, I like to show this diagram because there's a family relationship here. It's my family. My lab has solved the structure of the big sodium channel. My husband's lab has solved the structure of the little lamotrigine molecule. This is important, but it's one thousandth the size of this molecule. So I say size matters, and we've done the big one. Anyway, just a personal note. Okay, so this is the end-on view of this, and what happens is that lamotrigine molecule comes in here, and it sticks in the middle, and it blocks the channel. We think we know from doing molecular modeling and calculations where it is, and now we're in the process of actually determining where that is. Once we do that, we can make even better and more specific drugs there. So, drugs that must bind to sodium channels either stop them opening or closing or can keep them open to treat certain diseases. So lamotrigine stabilizes in inactive state, whereas lidocaine, a local anesthetic, binds and opens inactivated states. This is why you don't actually feel anything when you get a local anesthetic. I recently had a shoulder operation, and I've bothered the anesthetist enormously before going under, asking what all the local anesthetics were that I was being given. And it turned out they were all the ones we were studying, so I said, okay, they could go ahead and use them because I knew that what they did. But because they must be very specific to sodium channels in different tissues. And the reason that we can now help to develop specific pharmaceutical drugs is looking at the differences between them. So I said that there were 9 human sodium channels, labeled 1, 2, 3, 4, etc. But they all are found in different tissues and do different things. So NaV1.1 is found in the brain and causes epilepsy. Now, 1.2 is found in both the brain and peripheral nervous system, causes epilepsy and seizures. 3 is found in the brain and heart. 4 is found in skeletal muscle and causes myotonia. 5 is found in the heart and causes fibrillation and long Q3 heart syndrome, etc. This one's the one that people, and this one are the ones people are very interested in for chronic pain because they're found in many tissues, but they are very specific to chronic pain. So what we want to do is figure out a way that we can make something that will specifically affect epilepsy, but won't affect, for instance, your heart fibrillation. And we can do that now by looking at the very small differences. So these may be between 80 and 90% identical of all of these, but we can exploit the differences between them in different tissues to make very specific drugs. And finally, one of the things that was unknown until the crystal structure was determined was how these drugs got into the cell. People always thought they went through the top and through the selectivity filter and went into the central chamber. But it turns out they're not, this part of the top isn't big enough really for them to go through. But what surprised us when we solved the structure was to find these holes, the fenestrations. These are in the middle of the membrane, and most of the drugs that we talk about are very hydrophobic, and they can partition into the membranes and then go through the holes. But we now know that there's a big difference in the size of the holes in the open and closed state. And this may help us to guide and make new drugs that will bind only to the open or closed state because they'll only allow drugs of certain size to go through. So, to summarize, crystal structures have provided an insight into human disease, health, and the design of pharmaceutical drugs. The crystal structures show us the details of the inside and outside shapes of the molecules, and this can aid us in understanding what goes wrong in mutants that cause disease. The crystal structures show the differences between the open and closed states and are important for the design of drugs. Small differences in the amino acid sequences in different tissues means the drugs can be made that can cause fewer side effects. And in conclusion, this is our justification for being. We think that structural biology now can provide important information for the understanding and treatment of diseases associated with sodium channels and other molecules. And the most important thing is that these are the important people that have worked on this molecule over the past decade or so in my lab, including a large number of PhD students and postdocs, some of whom worked for years just to be able to make a milligram or so of the protein. We've collaborated with the people over at UCL on EPR studies and with people at Washington University in St. Louis to look at function, and at Harvard University also to look at function. And all of the work we do are at these very big instruments called synchrotrons that we go to in the UK, France, and Switzerland that have been very generous to let us use their facilities. And probably most important, and this is the advertisement part, but it's what keeps us going, is that the work on this has been funded by the biological and Biotechnology and Biological Sciences Research Council, known as the BDSRC, which is our major council for funding such things on human health. The Wellcome Trust, which is a charitable trust that's been very generous in funding disease-related studies. The Physical Sciences Research Council, which has provided us with new technology for doing these measurements, and the EU, which funded at least one of the postdocs in the lab to do some of the studies. And without the real dedication of all these people and the help and source of funding from all these sources, we would really not have been able to do this work, which we hope will now lead to things that can help both the human condition and develop new drugs. And I thank you.
Questions for Bonnie?
Yeah.
Yeah.
Now the channel is quite a large molecule.
Mhm.
The sodium ion is a very small ion. Why do you need such a
Yeah, that's a really good question. Why do you have such a big thing if you only want to let a very small thing through? Well, the tricky part is not so much letting it through. You could let it through. There are small antibiotics that let sodium ions through, but they're not regulated. The important part is that you need all the rest of the bits to regulate it opening and closing. So you can make a hole. Making a hole through a membrane isn't hard, but making a hole that you can regulate is hard. So you need all the bits to hold it together so you can, at will, choose by regulating it with toxins or drugs or voltages. So it's all that rest of the stuff is what it's there for.
Do they list the necessity for a post-seed box for the sodium ion transit?
Sodium ions go as long as the hole is open, they go with the gradient of sodium. So if there are more sodiums outside, which there usually are, the sodiums will go through as long as the hole is open. You don't need any energy source to make it go through. If, however, you let too many sodiums go through, there's another molecule, which we've worked on too, called the sodium potassium ATPase, which actually will send any excess sodiums outside, but that requires an energy source to force the sodiums out. It requires the hydrolysis of ATP. So sodiums can come in without an energy, but you have to push them out to get them out.
So in your animation, maybe it's too fast and fast. Could it be a possibility for the calcium to enter the pore? If there's no to come out again?
Yes.
Will there be a browning sort of motion?
Yes, in fact, I think I could show it to you again if you like. That they actually, they don't all go through. You may have noticed they kind of bounced around, and we had to wait for a long time for one of them to actually make it there. They go partially in, and then they go out, and then they go a little further in, and then they might come out again that side. There is ultimately a flow in the direction of the lack of sodium, so from the high sodium to the low sodium, but it's not a straightforward shooting through. It actually has a lot of back and forth and Brownian motion, as you say, that it goes through before it actually makes it all the way through.
Yeah, lastly, actually, I read the paper just before I came. In one of the papers, maybe it's not very good, it says that by a simple change in the
Very well noted, yeah.
So why is that so? Because the sodium and potassium will have calcium is
It is quite a different molecule. It's, let's see if I can find this very quickly. So that most any picture will do. Just need one that you can see, and okay, that's not a very good one. Great. Okay, these are sodium ions. Sodium ions actually have 4 water molecules around them, and this place in the middle is really quite big, and it can slosh around with waters around them, and the sodiums take their water molecules with them. On the other hand, if you change on these 4 residues, 4, if you change them and you change one position, you make it so it's not selective for sodium, but several different ions can go through. And if you change it to another 2 of the positions, you can make it so that calcium goes through. And calcium will go through, which is a different size ion, but it's unlikely to go through with all of its water molecules present. So the changing of the residues actually allows the complexation with the water molecules in the middle that then allow things to go through. So yes, it's quite a subtle difference, and that's one of the beauties of these channels, is that you do have calcium and sodium and potassium specific channels, but there's not very much difference between them. The potassium channels are quite a bit different because they're much narrower, and they can't, and so the other sodiums and calciums can't go through, but you can make a sodium channel into a calcium channel.
I don't know whether it's possible to explain to a layperson roughly what the process is of going from isolating the gene to actually producing the protein. How do you do it?
What you do is, as you know, the Human Genome Project determined the sequence of all the genes present. And so you can find the gene, you know what its characteristics are, of a sodium channel. And what you do is you make a piece of DNA. You either take it out of the genome itself, or you can make it synthetically, chemically, and you make that gene. And then you take that piece of gene and you insert it, in our case, into a bacteria. So E. coli, the very common bacteria. And when you put it in, you stitch it in the middle of the genome of the bacteria itself. And when the bacteria grows and makes its own proteins, it takes along this one by as sort of a rogue rider with it. And when it does it, it makes this. And if you carefully regulate it to downregulate how much of its own things it's making and make more of this, you can get it to churn out large amounts of this. And then we take the cells and we dissolve them up in detergents, and we isolate the protein and purify it. So nature has given us the way to make large amounts of these proteins. And that's part of the reason why you can't make the human proteins, is because bacteria don't want to make human proteins. So you need to make them in human cells. But these, you can make other bacteria proteins in E. coli just very nicely by just sticking it in there and hijacking what the cell is going to do anyway.
Yep.
So with some of these channels, there's like a mutation where there's only 3 of the aromatic ring, not 4.
Mm-hmm.
So how would you go about solving that problem? Because that's the first point of contact. If there's a hole there, there's a hole. You can't close it.
But if you know what the structure is, I'm just going to go to this one because it's easier to show it in the picture. Right. So this, what I haven't shown you further up, is up here, is what we call the cavity, and it's a bigger open spot. And in fact, up in here is where most of the drugs bind that are channel blockers. And so the point is, you can't necessarily change this because that's the person, and so you're not going to change what the sequence of the molecule looks like. But if you go up here, so up here, in a section up here, if you make something that blocks it by binding up in here, then you make it at least harder for sodium ions to flow unaided. So you're not fixing this, you're fixing somewhere else.
If there are no more questions, I'd like to do one final commercial advert for the last talk of Science Week, which is at 6 o'clock this evening in here. It's on the ethics and psychology of testing people in medical genetics. So is there any point in finding out whether people have these mutations that cause pain or remove pain if there's nothing we can do about it? I think in these cases there probably are things we can do about it, but some diseases you can test for, you can tell somebody they've got the disease, but there's actually no treatment. And Professor Jonathan Smith from psychology is going to talk about his research, understanding how people react to such information. So I'd just like to thank Professor Bonnie Wallace once again for a very interesting talk.